Dear all, today I am going to tell you about stem cell assays and techniques, and this is part 5. We have already seen the stem cell characterization by species characterization by karyotyping and with the help of chromosome banding and chromosome painting. Also, we have seen the isozyme analysis by using isoelectrofocusing and two-dimensional gel electrophoresis. Uh, in the last lecture on isozyme analysis, we have talked about electrophoresis, and this is zymograph, which is the pattern of isozyme distribution obtained either from chromatography or electrophoresis. In this, you can see that if this is A1 is one species, A2 is another species, and it is checked for one particular enzyme, then it has a single form of enzyme, and A2 has two forms of one enzyme, and if they are mixed, we get these three bands. This is how we can check where which sample is containing hybrid of two species. This is how we can check the lineages also, cell strains, and different cell lines. So these three are useful in that. Next technique is immunohistochemistry or antibodies to cytokeratins. These can be used to check the cell surface antigen and cytoskeletons also. DNA fingerprinting may be used for the same purpose, in which restriction enzyme digestion, polyacrylamide gel electrophoresis, and satellite DNA probes. These things are used, and these techniques are used for doing the DNA profiling. PCR of microsatellite repeats is done. So today I am going to tell you about the immunohistochemistry. These techniques detect antigens in tissue section by means of immunological and chemical reactions. This technique is highly sensitive and specific and can detect a wide variety of antigens in multiple species. We can check, we can see the presence of antigen in tissue sections, and the technique is available for multiple species. What is the unique advantage of this technique? That over other protein detection methods, that uh, it has ability to correlate the presence of antigen with its location in the tissue or cell. So technique can be followed in tissues, intact tissues, or single cell suspension also. And this is very important for study of cell function in normal and pathological tissues, because it is seen that in normal and in normal conditions, under normal conditions, position and presence of antigen is position and presence of antigen differs from the pathological tissues. So we know this, so we can check if the tissue has been taken from some diseased condition or it is from some normal individual. So first of all, what is the principle of the technique? Technique is same as for any other immunological techniques. Here you can see that this slide is coated with one particular antigen, against which there is one monoclonal antibody, which is in the form of conjugate, and with this enzyme is linked to this monoclonal antibody, and then it is treated with its substrate, and we get detectable product, uh, because either it is colored or fluorescent material, or it can be some radioactive material also, which can be detected, and this detection is directly proportional to the amount present of this antigen. This is a direct method in which this first antibody is carrying this enzyme. We have one indirect method also, in which this we use secondary antibody. This is primary antibody against this antigen. Secondary antibody against this antibody is known as conjugate antibody, because it is conjugated to enzyme. Again, we need a substrate, and we get a detectable product. Here in the protocol, first of all, we prepare our sample, and that sample is loaded here. After certain period of incubation, this is washed to wash off the excess material, and this antigen, if it has property to bind here, it remains on the slide, and rest of the things present in the sample are washed away. Then monoclonal antibody is used, and that will combine with the antigen, but there will be excess of monoclonal antibody in the form of enzyme conjugate, and that will be again after certain period of incubation, that will be washed off. Then when on the slide there is only this combination is present, then substrate is added, enzyme catalyze the substrate to make product, and that product is if it is colored, then it is visible, and we can see that. Same protocol is followed here. This is a longer procedure, and after addition of every product, every substance, or like antigen, antibody, primary antibody, secondary antibody, or conjugate, enzyme conjugate, uh, every time we wash thoroughly this slide, so that only this combination is present, remains stuck on the slide, and rest extra material uh, is not present. Then finally we get detectable product. So this was the principle. Now the protocol of immunohistochemistry. Here this protocol shows is given for the detection of antigen in tissue, and a section of less than 4 millimeters is taken, or cut, that tissue is cut into this dimension. Then it is fixed in formalin for 12 to 24 hours, while the time varies with various samples. उनके fixation में जो difference time लगता है, that will be given for tissue fixation. After fixation, this will be trimmed and embed in paraffin to make it hard. Tissue remains a soft material, and it is made into hard substance, so that its sections, thin sections, can be cut, and the size of this section will be 3 to 5 microns only. Then this section will be placed on siliconized slides. It is dried uh, at 55 to 60 degrees centigrade in oven for around half to 1 hour. Then finally it is stained. For example, in this principle we have shown that it is stained with a conjugate, enzyme conjugate. So it will this staining will be done, and before staining, paraffin should be removed, and section should be rehydrated, and finally we can see it under microscope. This protocol is shown here in this figure also. In xylene, yeah, sorry, this is a paraffin sample. Paraffin sample is this, but it is coated with paraffin. To make it hard, then after section cutting, xylene is used to uh, it remains in the xylene and xylene का concentration घटा दिया, then it is bring in ethanol. Ethanol का ethanol deep paraffin samples in ethanol paraffin is removed, and then we can clearly see the section, but uh, in ethanol it is dehydrated, as a result of which cells will be uh, shrunk. So to rehydrate it and bring the correct dimensions of each and every cell and the correct condition of the antigen, we need to rehydrate it, and various these things are known as washings, and finally it is in water, and then it can be seen under microscope. So this is how we can check the presence of antigens, which are species specific, lineage specific, strain specific, and their presence in the tissue itself can be uh, followed, and this is required to find out uh, uh, before starting our experiment. We need to know that on which sample or on which stem cell we are going to work. So this is all for today, and if you like my lecture, please subscribe it, like it, and share it.